share that Cayenne Cancer Health Initiative, the program they did last month to create awareness about breast cancer has led to a lot of people actually realizing the importance of early detection, and we say thank you to them for that. In the same way, we're also going to be appreciating another partner here, ICARE, which stands for Initiative for Community Advancement, Relief and Empowerment. In ICARE, one of the members of the advisory board for Cayenne Cancer Health Initiative is a partner in ICARE. Thank you. Another organization that is involved, ICARE is based in Shagamu, Nigeria. Another organization that is also a part of this webinar is the Nigerian Cancer Society, which is an umbrella body in charge of all the NGOs that conduct cancer advocacy. NCS coordinates everything in Nigeria that deals with cancer advocacy. And yet another partner is Fountain University Osogbo, where our guest speaker of today is a lecturer. Osogbo is in Nigeria also. Then we have another organization with us today, partnering with us for this webinar, ECCNI, which stands for End Cervical Cancer Nigeria Initiative. They are located in Kebbi State, and our chairman for today is the executive director of End Cervical Cancer Nigeria Initiative, ECCNI Nigeria, Dr. Ishaq Lawal. Thank you very much. And then we have the Medical Women's Association of Nigeria also partnering for this webinar. M1 is the umbrella body that coordinates all the female medical doctors and dentists in Nigeria, and M1 is in 36 states in Nigeria, as well as the FCT. Thank you. And then another partner with us today is Cancer Consciousness Initiative. And let's remember that CCI, that is the Cancer Consciousness Initiative, moderated the webinar sometime this year when we created awareness on bone cancers. Thank you so much for that. And we have the Medical Women's Association of Nigeria, Kaduna State branch, that has also been doing a lot as regards cancer. And we know that KCHI is in Kaduna, and so M1 Kaduna has a lot they've been doing with Cayenne Cancer Health Initiative. I want to appreciate everyone for being here. Thank you, because without you today, this webinar cannot go on. So I urge us all to stay put and listen to us as we discuss one dose at a time, current trends in research and development in cervical. So this is going to be our agenda for today. Okay, now we are on arrival and registration. Thank you everybody for coming. My name is Dr. Ifeinwa Morino KK. I am a senior resident in the Department of Community Medicine, Alex Ekweme Federal University Teaching Hospital, Abakaliki in Ebonyi State, Nigeria. And I'm also the Medical Women's Association of Nigeria National Outreach Committee Chairman. We're going to be taking some ground rules, please. So permit me to stop sharing this slide and then share our ground rules so we all know what is expected of us today while we are here. Thank you very much. Let me share the ground rules. Thank you. So for today, we have some housekeeping rules. Please expected to avoid noise in our environment as much as we can. So it's important we keep our mics muted. And whenever we want to speak, we please unmute our mic. And let's turn off our webcam during this presentation. Please, let's try to exercise patience and wait for our turn to speak. We're advised to take notes. There's going to be a question and answer session. And as you take your questions, please make it brief because others are also waiting to take theirs. We kindly ask us to rename our devices to have our name and organization because we're going to be having a report on this, and it will be important to have the names of those that attended as well as the organizations for future purposes. Please turn off our cameras anytime we have breaks. And we also advise us to avoid using this chat room for other personal conversations. So also a reminder to mute as much as we can when we are not speaking. There will be recording of our slides and this session, and we're also going to make them available in our website, www.kchi.org.ng. And we are going to expect us to give feedbacks and requests for participation certificates via the address that is being shown there. So please, we beg us to adhere to these housekeeping rules. And I know we are good citizens and we are good people who are going to adhere to these house rules. So thank you very much. So I'm going to be calling on the Executive Director of End Cervical Cancer Nigeria Initiative to give us the chairman's opening remarks. Welcome, sir. Dr. Ishak, welcome, sir.
Yeah.
Can we hear me?
Can you hear me?
All right. Yes, sir. Thank you, sir. You're welcome, sir.
Yeah. Good morning.
Morning, sir.
Yes, I want to thank all the participants for creating time to be at this webinar, and particularly thank the organizers for deeming it fit to invite us to be part of this. And it's really great. Listening to your introductions and the coalitions you're putting together it gives us hope. As you said, I'm a gynecologist practicing at Federal Teaching Hospital, Brini Kebbi, used to be Federal Medical Center, Brini Kebbi, and I'm very passionate about cervical cancer prevention, and that's why we formed this End Cervical Cancer Nigeria initiative. So listening to what you put together, how you have been able to bring all these organizations to come together and work toward the same goal is impressive. And listening to the theme for this discussion, which says one dose at a time, current development, research and development in cervical cancer is very apt. I suspect, without preempting the keynote speaker, that this theme was chosen to reflect the single dose, the migration from multiple doses of HPV vaccination to single dose, and try to tell us the research angle that led to this change, which is one of the big things regarding the possibility of Nigeria meeting the cervical cancer elimination targets. So my hope is that with all the stakeholders on this platform listening to the research, the presentation, we will migrate from our talk about single dose to single woman, because the ultimate goal is to reduce or eliminate cervical cancer in Nigeria. So we hope that after this single dose current development, we'll move to single woman, so one woman at a time, and collaborative efforts towards cervical cancer elimination. Because to eliminate cervical cancer, all the three prongs of WHO cervical cancer elimination targets must be implemented in synergy, which is vaccination of adolescent girls, screening of adult women, and treatment of all screen detected cases. So I'm not here to give a lecture, but just to welcome you. And if also, let me also use this opportunity to also inform you of the stakeholder summit on cervical cancer elimination that we are planning for the month of December. It's about to start. The pre-summit is starting next Monday, and the summit itself is 5th and 6th of September of December. And I invite all of you to be part of it, because the goal of End Cervical Cancer Nigeria initiative is to see that Nigeria meet the cervical cancer elimination target, and that cannot be done by an individual nor an organization, no matter how big you are. It requires a collaborative effort like what KCHI is doing here now, bringing everybody together to collaboratively work together, brainstorm, and march forward towards achieving cervical cancer elimination. So I enjoy you to please listen, as was admonished by the moderator, Ifeanyi, that we should also take note, that we should not only, it's not about listening alone, we should take note. And as we are taking note, we should make mental note also of possible actions that we are going to take, because it's not just only taking, listening and getting ourselves educated. The lessons learned, the knowledge transferred here should be transferred into, to be translated into action for it to be an effective project. So once again, I welcome you and wish us a very fruitful webinar. Over to you, moderator. Thank you.
Okay, thank you very much, Dr. Ishak Lawal. Yes, you're the Executive Director of End Cervical Cancer Nigeria Initiative, ECCNI Nigeria, and also you work in the Department of Obstetrics and Gynecology, Federal Teaching Hospital, Birin, Kebbi State. And that is why I also said that ECCNI has its headquarters in Kebbi State. Thank you very much, sir. Yes, you've shared the three prongs. And so we're going to, just like you told us, listen to what our guest speaker has for us. But before I call on our guest speaker, Dr. Nusra Bolatito Afolabi Balogun, who is a professor and head molecular biology and genetic diversity laboratory, biochemistry unit in the Department of Chemical Sciences, Fountain University, Osogbo, Nigeria. And she also partners as the principal investigator, African partner, Cambridge Africa Alberado UKRI Initiative Project. Please permit me, everyone, to read her citation before she comes on board. Thank you, and let me share this while I'm doing it, okay? The citation of Afolabi Balogun Nasru Bolatito, PhD. So I'm trying to get it on so that we can also follow. She holds a doctorate degree in biochemistry from the Ahmadu Bello University. Can we see it? Hello?
Yes, ma'am, we can.
Okay. Nasru Afolabi Balogun holds a doctorate degree in biochemistry from the Ahmadu Bello University Zaria. He started an academic career in her alma mater in 2006. She is a distinguished professor of clinical and molecular biochemistry, Fountain University Osogbo. Nasra is a member of the International Union of Biochemistry and Molecular Biology and the Southwest Coordinator of the Nigerian Society for Biochemistry and Molecular Biology. She is the principal investigator / founder of the Molecular Biology and Genetic Diversity Research Laboratory and the African partner of the Cambridge African Alberado UKRI ODA initiative, termed Building Capacity for Early Diagnosis for Cervical Cancer in Africa. Her group specialized in using biotechnology and genetic engineering to explore disease diagnosis, signal transduction pathway, and translation research with bias in genetic disease, such as sickle cell disease and gynecological cancers. With vast experience, both internationally and locally, she is a well-traveled academia and distinguished researcher with collaborators within the African continent, Europe, United Kingdom, and the United States of America. She holds distinguished fellowship and grants, notably the Camp Africa Eldorado UKRI ODA International Union of Biochemistry and Molecular Biology Mid-Career Fellowship, the Federation of European Biochemical Societies Cancer Research Fellowship, MacTor Foundation Moros Integrated Cancer Research Grants. As an academic mentor to younger generation, she is conscious that the future of younger generation is dependent on what is given by mentors. Thus, she thrives to empower herself with knowledge through continuous training workshop and fellowship to train and retrain to ensure she would not be letting down her mentees. A dedicated mother and wife, she holds family ties and high esteem with unbeaten flair for her hometown of Kwara State. Join me to make welcome our guest speaker for today, Dr. Nusra Bolatito Afolabi Balogun, a professor and head molecular biology and genetic diversity laboratory, biochemistry unit, Department of Chemical Sciences, Fountain University, Osogbo, Nigeria, as well as the principal investigator, African partner with Cambridge Africa Alberado UKRI Initiative Project. Welcome, Ma, as you take us through the topic, one dose at a time, current trends in research and development and cervical cancer. Thank you very much, Ma, and welcome.
Thank you very much. It's a pleasure, and maybe a quick apology. I will be giving this webinar from an hospital bed, but then it's fine. It's just the situation I caught flu. So, but otherwise, everything will be fine. I don't know, can everybody hear me? My voice is a little bad. Sorry, can you hear me?
Yes, ma'am.
All right. So, do I have the permission to share my slide? Thank you very much. So, it's a pleasure to be here today. I'm Nusra, and I will be giving a talk on one dose at a time, advances in cervical cancer research and developments. And I will be looking at it from the point of view of exploring through a breakthrough in vaccine treatment and policy. So to do this, I would start with just briefly. I normally say when I'm giving talks on cancer that I do not have the expertise that it takes to talk about cancer because I only know what cancer looks like on paper. But then, after so many years of looking at this, I could say that cancer is a dysregulated or dysfunctional growth when the body loses its capacity to stop growing, because our bodies are made to grow, the cells in our bodies are made to grow and die. But in cancer cells, they lost this ability to do so, and hence they continuously grow. We have several types of cancer, blood cancer. So because cancer is a tissue problem or a tissue disease, you can have cancer of almost everywhere. Cancer of any cell, any part of the body that has cells can be cancerous. And but for the purpose of today's lecture and from my own area of niche, I deal specifically with cancer of the cervix, which is called the cervical cancer. And before the cervix begin to grow uncontrollably, it requires an infection. In most cases, 90 to 99% of cervical cancer cases are caused by the human papilloma virus. And what normally happens is after this infection occurs, it causes what the clinician calls dysplasia. That means there is a dysregulation in the morphology of the cervix. And there are several types of HPV which we are going to get to, but usually this is the way the cervix looks like. So in the case of normal cervical, the cervix, it looks in this form, and with infection, persistent infection, then there is what becomes cancerous. As I mentioned, there are several types of HPVs, that's the causative agent of cervical cancer. We have the alpha, which is the one that has been implicated in HPV-related cancers. And with HPV infection, we could have warts. Warts looks like a ball. It's more like a lumpy, watery growth, which could be in any part of the body. I must mention, apart from the cervix, HPV or human papilloma virus has been also implicated in head and neck cancer. It can be associated with genital warts and other form of cancers. And so that's what. Now to the current status of cervical cancer globally. I want us to look at it globally. Generally, we have cancers in every part of the world. There is no part of the world that is isolated. There is in Africa, Asia, America, and Europe. And this is just a cross-section of what the population looks like. And if we look at this chart, this is like a global map of what's the distribution of cervical cancer. And we will see that it is really bad in Southern Africa. And yes, the Nigerian population could be seen to be in the range of 16.7 to 25% of this. And this is looking at the mortality and morbidity. And here is the East African population taking its lead in Africa. And we have West Africa somewhere. Yeah, this is West Africa. So, and as shown in the map, Southern Africa has the highest population. But in terms of mortality rates, Southern Africa still has their own. They're still leading in the African continent, while Nigeria follows closely. Hence, when this topic came to me, what I did was to look at the study on the data available in literature. I'm a researcher, so I go back to literature. So my search word was HPV, the causative agent, cervical cancer, and genetic study. And when I look at it without any modification, I got over 8,000 papers, publications in this field. Then when I used just cervical cancer in Africa, I got less than 500. But when I included Nigeria, our own country, I got less than 70. While when I say genetic HPV, cervical cancer, gene expression in Africa, we've got just 2, while we do not have any in Nigeria. There is no study on the genetic expression or genetic study in cervical cancer in Nigeria. Thus, I will take us to what the globe has been doing about cervical cancer, understanding the cancer itself. So to do this, I will be looking at the work of Charavanti et al. And in their paper, what they did was they asked the question, are there conserved molecular subtype? Are there molecular subtype of cervical cancer that's influence the expression of this globally? To do this, they looked at the cervical carcinoma, squamous carcinoma, and they had looked at 5 cohorts, including the Atlas of Cancer Genome, the Cancer Genome Atlas. They had an African country, which is Uganda. One, like I mentioned from the map I had shown us, that Eastern Africa has high population, followed by the Southern Africa. So Uganda is one of the countries that had ever done, one of the 2 countries that have genetic study in cervical cancer. So Uganda was involved. We have Oslo and other countries. And the basic analysis they did was they ran RNA-seq methylation, typing, tumor clustering, pathway analysis, and among others. Then they did mutational analysis, copy number, and the rest. These are core molecular biology analysis to give an insight into what is actually happening in cervical cancer cases. So their findings, they were able to establish there are 2 clusters that, yes, there is, as mentioned earlier, there are several types of HPVs, but 2 major types of HPV has been implicated. That is the belief. And it is based on this belief that the vaccines were developed. So they looked at HPV positive squamous carcinoma individuals from their 3 cohorts. So 3 cohorts, US, Europe, and Uganda representing Africa. And at the end of the day, they were able to establish that there are 2 cohorts, the C1, which belongs to the alpha clades. This will be a little tautologous because we don't go in there. But then the clade 9 belongs to what we would know as the HPV-16 group. It's not only HPV-16 that is in that group, but the most common one that we would have known in Nigeria would be the HPV-16. Then there is the C2, that's the second cluster, which belongs to the alpha 7. I had mentioned earlier, there is alpha, beta, and different type of cervical papilloma virus. So when I say alpha 9, that means the alpha papilloma 9. Now, and so they also looked at the clinical significance of these 2 groups, and they discovered that the C2, which exhibit overall poor prognosis. They exhibit that the survival rate is very poor when compared to 79%. So they had about 52% survival rate when you compare them to the 79% that was observed in the C1, that's in the cluster 1 group. Again, this group, like as being mentioned, belongs to the alpha 7 subgroup, and they were able to liken their observation with the aggressive nature and worst clinical outcome with individual infected with the HPV, let me use the HPV-18. Hence, they went ahead to look at the association of these 2 clusters with the prognosis in an independent cohort. Initially, they had done their own analysis, and then they went into another cohort, that's into a population, more like a field study, and they were able to establish that the prognostic value in these two clusters is not dependent of the predictor of the survivor. But they also noticed that the C2 clusters has a very poor disease-associated, disease-specific survivor. So, and the overall survivor is also very poor. Hence, they were also able to establish that these studies was carried out in chemotherapy naive patients. That's patients that have not gone through any form of chemotherapy before. So they just started this and they studied and they discovered that the cluster 2 individuals had poor prognosis and response to treatment. Their study also looked at the relationship between cluster and between these two clusters and the previously identified, that's the atlas. So they were, because theirs was an independent study. So they compare what they had with what the atlas has. So, and the tumor atlas they were able to confirm that the C1 clusters were very close to the mRNA C2 keratin I. That's the one that has I keratin clusters in the TGA. And the C2 they have strong association with those with low keratin component. So that means what these this cluster classification they did is it has been recognized in the tumor atlas. And what they call cluster 1 were those with high keratin clusters, and the other cluster 2 are the low keratin clusters. So, and they also realized that the C2 tumors, that's the ones they were classifying as C2, were the ones that correspond to the epithelial mesenchymal transition in the atlas. So they also look at their prognosis, they try to look at prognosis in terms of, that's genomic prognosis in terms of these different clusters. And they discovered that there were 4 mutation signatures. The first being in the comic signature 1, and this is the age-related mutation. That's the mutation is observed in people that are showing maybe late age. That the aged ones has a lot of comic signature, while ApoPec 2 and 13 were seen in the middle age. So these were ApoPec-related mutations. And the last mutation they observed is the ApoPec, is the comic signature 5. And these are of unknown origin. And they didn't see any difference between cluster 1 and 2. They identify also some new mutation genes, that significantly mutant gene, and 21 of these genes were not previously associated with cervical cancer. And they discovered that the phosphoinositol kinase and EP were associated with C1, while STK and NF were associated with C2. And this led them into studying the pathology or the signal pathway of the C2 tumors. And they were able to see that the C2 tumors stimulate the pathway alteration. And this pathway alteration is usually associated with the YAP1 gene. This YAP1 gene is responsible for protein expression, and any aberration in this is responsible or has been found to lead to aggressive and metastasis. Hence, this explains why individuals with C2 mutation or C2 infection, or going back to the beginning, and individuals with infected with HIV, HPV-18, the the cancer associated with it progress aggressively. The cancer progress aggressively, and it's metastasized fast. And that explains the reason why there is poor prognosis with individuals associated infected with the infected with HPV-18. So I hope I'm not doing a lot of Cambridge English, but then that is what has been done in the world. And in Africa, the Uganda case. So the Uganda case, this is the paper I used to review it. It's a 2021 paper, and they asked the same question, trying to understand the epigenomics and transcriptomics of cervical cancer of different HPV cases. And to do this, they hypothesized that HPV client may push epithelial cells to replicate at two ends, with Clyde-7 driving a less differential phenotype. And they use the Uganda population, like I mentioned, and they look at the prognosis of this. They use the atlas, the tumor atlas, as their reference, and the human genome. And the outcome of their research showed that there is a difference in the HPV, that different HPV clients have different morphology. The DNA methylation, the promoter, the enhancer, histone markers, and gene expression, as well as the pathway of dysregulation, were different depending on the HPV strain that the individual is infected with. So they had a map to show the population of the various HPVs that were detected in their environment. And as a summary, they have 9 HPVs were responsible for 54% of the HPV in that population, which is led by HPV 16, and 82, then 71. As against the general belief, or what led to the design of the HPV HPV vaccine, the general belief is that most of the HPV cases were associated with 16 and 18. But the Uganda work, or the Uganda research, has shown that 46% of the HPV in the population or in their circulation was not covered. Hence, vaccinating the Uganda population with the current available vaccine would leave on the average of 46% of the population uncatered for. So they went ahead and looked at client-specific histone marker landscape. The heat map, don't let us look at the heat map, but what I would just wanted us to pay attention to is, okay, they carried out their own with HIV and looking at co-infection, but then they were able to look at 4 stages, and they were also able to classify it into 2 clients, 9 and 7, but there is a little population that is in another client, and methylation also occur in different months. So this is an average of what they have been able to do, and they were able to identify 4 clusters as against the general clusters that were done, that the 2 clusters that were identified in the Chakravanti group. So they had 4 clusters, and the tumor grades were graded into 3, and they had more classes of HPV. Histologically, they were able to identify 4 different histological profiles, that they had the squamous adenocarcinoma, then adenosquamous cells. And from their cohorts, they were able to establish that aside HPV 16 and 18, 45 is also on the run or on a large scale in their population. They also find out that they have the Clyde-7, which I had explained, and liking with the TGS earlier. They were able also to say that most of the Clyde-7 are squamous carcinomas, and they found out that some of them, the HIV status did not really influence or have effect on this. As they went into decoding what actually is happening in each of these situations, they were able to identify mutation in 7 positions. 7 different mutations occur, and these 7 mutations were in different genes, but of major interest to, after deep studying of the work, what we capitalize or what we are looking at is mutational changes that is occurring in most of this. And we discover that they discover that the mutation changes were associated with the APOPEC gene. I mentioned from the earlier, the Chakrabarti group, that they also implicated the APOPEC gene. So this entire 7 mutation occur within the APOPEC mutation, and so we decided to look at the APOPEC gene, what actually happened in APOPEC, and we realized that APOPEC is just a demethylation, just the removal of a methyl group, of an amine group in the situation. And this now leads into modificational changes that could result in either more new epitones being formed, or you have high tumor modification, so mutation. And this is just a schematic representation of what happened when there is APOPEC mutation. And that is what, so it depends on the degree of APOPEC mutation. That's what determines the aggressiveness of mutation that occurs in a particular patient. And as we have said earlier, the HPV-18 individuals progresses more aggressively because there is more APOPEC mutation involved in their cases. Now, this knowledge was also used as a guide in diagnosis, prognosis, and therapy. And they have seen that if there is APOPEC mutation, they could use this to do immunological intervention. They could also use this, it has been used in breast cancer therapy to address immunological endocrine therapy, that in a situation where the APOPEC prognosis is used, and they are then used to do the upregulation of the epithelial endocrine reticulum. It has also been used in lung cancer, ovarian cancer, bladder cancer, and adrenal cancer. Hence, the study that is currently going on is to look at APOPEC and cervical cancer. To do this, we try to look at APOPEC activity as associated with viral infection, because cervical cancer is caused by a virus. So we looked at the mutational changes that is happening in that, and how HPV triggers mutation immunological response through the interferons to increase APOPEC protein production. This usually results in loss of p53 activities, thereby increasing APOPEC expression and mutagenic capacity in cancer cells. We have also been able to look at literature that the expression of the oncoprotein, that's the proteins that is expressed by the HPV virus, has also led to expression of some of the APOPEC family members. And this usually leads to persistent, so it subdues the immunity of the patient such that the APOPEC protein is produced more, mutation increases, and this leads to increase in the production of mutated cells, hence progression to cancer. So in summary, we are trying to look at APOPEC as an underlying protein in the pathogenesis of cervical cancer through HPV restriction and accumulation of mutagenic cells. And to do this, we propose to look at APOPEC's signature, what is the profile in the Nigerian patients. So we are trying to look at APOPEC signature in the Nigerian population. We are trying to determine the relationship between APOPEC detection and the carcinoma of the squamous cells, and we're trying to link this to prognosis and diagnosis. This is just a chart of what we intend to do, or the way we are looking at it, to look at the proteomics using MS, then transcriptomics, then we are going to do a selection of the group to look at. We have a research to look at, aim to look at the biomarkers that might be associated with this, so that it will be a better targeting for, so as to have a better targeting position. And we hypothesize that if we understand the omic pattern, we would be able to get, and we know the signal pathway, just like I've explained the signal pathway and everything as I've explained earlier, then the Nigerian population would be able to have a better understanding and a better therapeutic target. So the work so far that we have done, we have looked at immunological or protein profiles of some of our of the samples that we have, and we discovered for individuals with various type of cancer, various type of HPV, the protein concentration differs. This is just normal biochemical extraction and quantification. Hence, we decided to run an SDS just to look at the protein, evaluating the proteins. And we discovered that, yes, indeed, in different individuals, there is a difference in protein profile. And for those, some of them actually express a differential bound. And this differential bound is currently being analyzed for the content, so that, yes, it is possible to get a biomarker that can be specifically used to target the Nigerian population. This is a genotyping profile that we're also doing in our lab. This is more like a point of care kind of diagnosis using ordinary cervicovaginal fluid. And these are ready-made enzymes that can be used at room temperature, at a little, just, we are still working on it, but then this is able to differentiate between individuals with different type of, so in this case, we have more of them having HPV 16 and not HPV 18. And that's just what this is. We are also using qPCR to genotype the population to be able to determine how much of HPV 16 or 18 do we have in the population. We are also using bioimaging. My lab has a confocal microscope, and we are currently using the microscope to look at the HPV profile, or the HPV differences in HPV morphology of patients using the cervical vaginal fluid biopsy and other samples that we have been able to lay our hands in the lab. So the way forward, from what I have seen or what I have shown earlier, I would say the Nigerian population has zero data on the genomic status of the cervical cancer cases we have. Hence, if we want to really get to the roots, or we are committed to, because one thing I do say in some of my talk is cervical cancer should not kill anybody. Unlike most other cancers, it takes on the average of 10 to 15 years before an individual moves from the infection itself to full-fledged cervical cancer. Hence, we should enhance genetic study to be able to understand. I know a lot of what I had presented looks like gibberish, but then it gives them, it's more like knowing exactly what is wrong with individuals and being able to therapeutically target that. So we are proposing that there is a need for us to be able to study the African population just the same way it has been done in advanced situations. Let us be able to cluster our cervical cancer cases. The preliminary work I showed you in my lab works with both FFPE sample, fresh tissue sample, CVS, and everything. So we are currently working on getting more of this together so that we can genotype, run sequencing analysis, and be able to give an insight into the present situation of the type, the prognosis, and the propose a potential therapeutic target for the Nigerian cervical cases. So our funding has been from the laboratory. This is mainly from my institution and the Cambridge Africa Alborada, and more recently, the UKRI OD Innovative Grants. Recently we got the phase 2 again, but yes, so we have phase 1 and phase 2, which we are working with. And I'm not out there so much, but I can be found on LinkedIn. Yes, my LinkedIn link, I'll add it to this. I have an OH, Google Scholar, and of course, ResearchGate. I can be found on those platforms. My acknowledgement goes to my entire team, my collaborators from Cambridge and BioRTC, and the entire team member in my laboratory. Thank you for listening, and thank you.
Wow, thank you very much. Thank you very much, Dr. Nasir Balogun Tito. Thank you so much. Please, everyone, can we appreciate her for this wonderful presentation? And in a special way, the fact that you're even on an admission bed and you were able to give us this with your lovely voice, thank you. So can we take questions, please? If you have questions, drop on the chat box. All right. I expect to be seeing us give claps. I expect to see electronic claps because she really showed us her stuff. Genetic biotechnology. It's not an easy one. Thank you very much, ma'am. Thank you very much. All right, our questions, please. All right, everybody is appreciating you. Thank you for the presentation. It was elaborate. Yeah, for me, something stood out for me. Can we hear me?
That's fine.
Okay, the fact that she shared with us what she is doing. And then it's alarming that in Nigeria, we actually have zero study when she used the keywords that had genetic markings. It means we have a lot to do. I want to appreciate you for even pushing this forward and for being there for Nigeria in this. I also like the fact that she talked about the alphabet protein and its role in the pathogenesis of cervical cancer, HPV restriction, as well as the accumulation of mutations, which led her to now bring up this proposal that she's working on. Thank you, ma'am, for that. So, do we have questions? Okay, thank you so much, Prof, for such an elaborate lecture. To what end is this research? A question, ma'am. Someone is thanking you and then asking, to what end is this research? Is there a possibility that there could be more HPVs in the future? And would there now be newer vaccines per time?
Okay, so to what end? So let me say the first thing or the main goal of our group is to establish a genetic profile for the Nigerian cervical cancer cases. We are gradually collaborating we're spreading from the south to the north. I can see Dr. Mariam on the platform from Kano. So to what end we are trying to generate a genetic profile for the Nigerian cervical cases. And when I say genetic profile, I went in depth despite knowing that most of these, except people in biotechnology, would not really go. But then I just went this deep to show what the world globally, what is being done, why and how they came across the vaccine for horse. They studied their population. And if we are able to look at the difference between the US case, that's the one at the Jakaravanti group, and that of the Uganda case. The Jakaravanti had 2 clusters, but Uganda is saying no, it is 4 clusters. By the time we come to Nigeria, we might see 3 or 7 clusters, looking at the diversity of our own country, the strength, the population. So we need to be able to profile our country. I call it profile because profiling for us in genetic engineering means you should be able to know where do who belong, who is the grandfather, the great-great-founder of who. So that is where we're trying to go. It's not cheap, but then we are open and we're collaborating. We're looking for funders. And let's say we are getting some, but we are trying to profile the Nigerian population. And when we are able to do this, even if it is just showing in hundreds of patients, in tens of patients, that there is diversity, that will stimulate a greater network. Yes, we've been screening. I know a number of screening activity going on. People are trying to do the genetic analysis, but it's not cheap. To do ordinary O exon sequencing per sample is about 350 euros. So 350 euro for only WAS. Now we need to do RNA-seq. We need to do so many other things to be able to attain what these people what I have shown you. But yes, there is no one is an island. So if we have our preliminary data, this will stimulate greater funding bodies. If we're able to get samples from 3-4 locations and we are able to push a little data out there, it will definitely enhance the funding so that we can be able to do a larger work. So.
All right, thank you, Ma. Yet another question for you, ma'am.
Yeah.
Besides funding of research, what else is needed for a smooth research in Nigeria?
One thing that is very important is equipmentation. And that's one of the things my lab is doing. I mentioned briefly that we have a confocal microscope. That is, I think, the second in the country. We have one in BioRTC, our collaborating institution. So with the bioimaging system, we're able to do immunological studies. We're able to see pattern where dysregulation. We're able to see specific protein modification. Aside that, we have an established tissue culture where we can grow our own cells. So I would say one thing that is we cannot shy away from to be able to do research in Nigeria is equipmentation. That will still be under funding because if you have the fund, that's when you are able to buy the equipment. And I would say another thing which people still say, dedication and honesty. Yes, dedication and honesty. The passion is also important because I could get the fund and do some other things. I could get the fund, get the equipment, but if the passion is not there, you will not be on the bench. I am happy and proud to say, even as a professor, I'm still on the bench. I still hold pipettes. So.
Okay, thank you, Ma. And there's a question here that says, how do we get to make vaccines in Nigeria where the technology to make them is not available?
Okay, so I'll use the title of this talk, One Dose at a Time. What vaccine do we want to make? A vaccine is made targeting a particular protein, a particular gene. We don't even have the profile. We don't even have the profile. So during this study, I was able to establish, and with some of our collaborators in IFE, that yes, we have HPV-16, we have HPV-18, but there is HPV-35. Uganda is saying they have 45, but we have 35. It's an ongoing work. That's why I'm not giving the details. But there is HPV-35, maybe only in the southwestern Nigeria. Maybe by the time we come to Abuja, by the time we get to Kano, by the time we go to Zaria, maybe these values will change. But we definitely need to genotype our HPV. Is after genotyping our HPV, we need to do a sequence to be able to know what are the main gene dysregulated, what are the mutational changes, and how does this mutational changes transcend into histone marker differentiation, protein modification. Is when we know the modified proteins, that we are able to then start thinking of vaccine. The initial vaccine were targeting the E6 and E7 oncoproteins, because they had looked at the points of protein modification, and that is what the vaccines were targeting. But if we do not have, like what they showed in Uganda, 46% of their HPVs were not catered for. That means the protection is only over 54%. Even if they are going to, that means they need to now design or understand the morphological changes, so signal transduction pathways. And it is when they know these pathways that you are now able to say, okay, I want to inhibit this particular pathway. Sorry, I'm a biochemist, so I'll talk more about pathways. So the first thing we need to do before we start taking a vaccine in Nigeria is we must understand what are the dysregulation in our cancer cases.
Okay, thank you, ma'am. Someone is saying no study in Nigeria. That's striking. What can be done about that?
Study, collaboration. I would say, as a researcher, one of the greatest challenge I've had is collaborating with the medics. Our collaboration is not strong enough, so we should build on it. We should get our, I'm sorry, my flu is now very productive. So we should get our doctors more involved in research. They have the patients. We possibly have the skill and understanding. We could sit down together and talk about how these things can go. To be able to do a significant study, sometimes you will say you will be targeting about 130 cases. How do you get 100? Or sometimes 200 and something cases. The Uganda study was 313. I'm sure to get 313 samples in Nigeria is going to be like a hell. I'm saying this from experience. So we should get more collaborations. The clinicians should get involved. Let's strengthen this research. Let's be able to fight this case together, write and push our findings out there so that we can attract more funding. I know people that have been doing HPV screening. I know people that have been trying to do genetic study, but our papers are not there. Our papers are not there. And if our papers are not there, we would not be able to attract funds. So we need to do more.
Okay, thank you, Dr. Miriam, thanks. She says to buttress Prof's points, genetic analysis and profiling will open up avenues for precision medicine, that is, individualized patient care. Yeah, I want to agree with you on that, Dr. Miriam. Thank you. And there's also another question here. Well done, man. Please, how can my hospital collaborate with you? Someone else had also asked, before this one, how can hospitals collaborate with you?
So we are open to collaborations. In the link here, I have the links for, and the organizers can definitely share my link, or if you go on, if you just drop my name on